The overall aim of this procedure is to graft a dorsal neural tube explant from a quail embryo into a staged matched chicken embryo, so as to trace the migration and fate of neural crest cells. This is accomplished by first tearing a small hole in the host chicken embryo's vitelline membrane to access the neural tube. The donor quail embryo is then removed from its egg to prepare it for graft excision. In the next step, the region of interest is excised from the host embryo's neural tube. A similar region is then excised from the donor neural tube for grafting. Next, the donor neural tube is transplanted into the host neural tube. Finally, the chimeric embryo is incubated to the desired stage for detection of neural crest-derived cells in the organ of choice. Ultimately, the migration and differentiation of donor-derived neural crest cells to various locations in the host embryo can be visualized through immunofluorescence microscopy.
Visual demonstration of this method is critical because the microdissection and grafting steps require very fine motor skills, making them difficult to learn.
Begin by washing any debris from the chick eggs with tepid water. Arrange the eggs horizontally on a tray and then mark the sides facing up with pencil to denote the region where the embryo will be localized. For ex ovo preparation of donor embryos, incubate the quail eggs blunt side up. Next, place the eggs in a 38 degrees Celsius humidified incubator and turn on the rocking function. After the egg